What have we gotten wrong about genes in the last couple of decades?
Well, I guess, I started with the traditional idea of how biology works. And I'm going to put it simplistically, but I don't think it's a gross distortion to say that probably since the beginnings of molecular biology and modern genetics, which is, the 1950s, Crick and Watson and DNA, um, the idea has been that life works because it's a program that's encoded in our genome and is just sort of read out. And it's read out to build us, and then it, continues to be read out as we go about our daily lives. So it's an idea that information in biology and causation actually flows from the bottom up, from the genes upwards. And, that made a lot of sense at the time because the genome is this string of information that kind of looked at the time like a string of computer code like you got on magnetic tape in those days. And I remember those days. The computer code was encoded on magnetic tape, so it looked as though this was how things were working. And that picture got us a long way in terms of understanding biology as a kind of information science, where there is information that's, it literally is encoded in DNA and is read out to make proteins, and that information is what's inherited. So it's not as though that picture was totally, wrong or fantastical, but it's not enough. And not just that it's not enough, but it gives the wrong impression of where the information is coming from, where the organization in biology is coming from. So, that was the traditional picture. It was kind of encapsulated in Francis Crick's central dogma. I mean, just the fact that it was called that sort of tells you everything. The central dogma of molecular biology, which Crick was quite careful about explaining, was really a statement about how this encoded information in a genetic sequence, how it moves around, how it's used by the cell. Basically, that it's used to, first of all, make this intermediary molecule RNA, and then the RNA molecules go away and are converted by a big protein complex called the ribosome into proteins. And all of that is fine. But the sort of implication was, and I think the way that message has tended to be received is, once you've got proteins, job done, because the proteins are little machines that somehow put us together. And that's not the way it happens. And it's really something that I think has only started to become clear over the past 2 or 3 decades, not coincidentally, since the completion of the Human Genome Project. So that old picture gave the message that all we now have to do is to decode the genome, and we've kind of got it all. We've got the answer to how life works. It hasn't turned out that way. We've decoded the genome. We've decoded now, tens or maybe hundreds of thousands of genomes, and we still don't fully understand how these complex systems of cells and of organisms work. And I think it's become clear not only that the information isn't all going to be found in some encoded form in the genome, but that actually every step of the process is slightly different, or sometimes profoundly different, from what we've learned. So that's really what I set out to do in the book, to step through those processes kind of at ever larger scales in the biological structure of the organism, to explain how things have changed in the past 2 or 3 decades. And even at the level of genes, there's been quite a lot of change.
I love it. I love it. So well explained. Because it's so intuitive to assume that the causality is from the bottom up, right? I think, and we'll get into causality will be a kind of one of those themes that goes throughout. It's just such a weird concept to think that it could be any other way. So what is the right way to think about what's happening there if the information is not coming from the bottom up? If genes do specify proteins, and proteins are machines, and point out where any of this might be incorrect or where more nuance is needed, and then proteins kind of build the rest of the organism. What's wrong about that specifically?
I guess my real answer to what's wrong about that is first of all that is not what modern molecular biology shows. So it's an empirical answer, and I was really keen to make that the guiding principle of the book. I didn't want. I'd seen there have been for years and years actually. Voices who've been saying, this isn't the right picture, and they've tended to come from a slightly abstract or slightly sort of philosophical position about what organisms are and what biology can be. But I wanted to root it in what we have found out at the lab bench in terms of the way these things actually work. And so
Yes.
I argue that it's that alone that compels us to think of a different kind of narrative. But, when you think about it too, building an organism by that bottom up way would be a terrible way to do it. It would be an insane way for, let's put it this way, for God to plan us. Because organisms are not like that. They cannot work like machines. If they worked that way, it would require every little bit to have to be in the right place at the right time, like a piece of machinery with billions of cogs, each one of which has to fit in the right place and has to turn. And if one of those is out of place, then the whole thing falls apart and stops working. You're never going to get a complex mechanism to work solely from that way. Plus, the nature of the biological organism is not the nature of a machine in the sense. So in the following sense, for example, if the idea was that the genome tells the organism everything it has to do in any sort of circumstance, I mean, how is that ever going to work? Every organism, and particularly complex organisms like us, every day encounter circumstances, situations that we have never encountered before and that our ancestors have never encountered before. It would be absurd to imagine that somehow there's a kind of lookup table in the genes for what we
Right.
should do in those circumstances. That is just not going to happen. We are, as all organisms are, we are making use of information from outside all the time, and that information, that context, needs to be what is governing our decisions. And so we need to have some way of integrating that information. It's a familiar idea to neuroscientists, that somehow our neural circuitry integrates information and comes to a decision about what we should do. But actually, that same principle applies all the way down the biological hierarchy to the simplest living organisms, to single cells.
Yes.
They too have to be able to make contextual decisions. So it can't all come from the bottom up. There has to be some way. Ultimately, I talk about it as there has to be some way to ensure that biological organisms are genuine agents. They're not just automata programmed by their genes.
Yeah, really well said. The expression that I love for this is biology is cognition the whole way down. This is sort of Michael Levin's work and Anna Chaunica, and it's just such a beautiful way of understanding biology. And it's just, I think you can't help but not get goosebumps when you read about it. You're like, wow, that's just simply so much more marvelous than I ever could have expected. After finishing this book, what do you think is the right way to think about what genes do? Because I thought that was, you go through a number of different pages and chapters of even just trying to define what a gene is very hard. Because this whole one gene, one function is completely wrong. This one gene, one protein, very wrong. So what is the right way to think about what a gene actually is and what it does?
Yeah, well, that's a discussion that I wanted this book to start. So, I'm not by any means convinced that I have hit on the right way to think about genes, but there are some ways better than others, I think. And it's certainly. I mean, it's not even the idea anymore that the gene is an instruction for how to build a protein. It's an instruction for how to build various proteins on the whole, particularly in our bodies, because the gene, encodes possible proteins of which several might be made, sometimes in different tissues, because we now know, and we've known since the 1970s, that when that information gets translated, get transcribed, I should say, into RNA, into messenger RNA, before it then gets converted into a protein, generally it's chopped up and edited, and bits are thrown away and the other bits are stitched together. And you can do that stitching in various different ways. And it does seem to differ very often in different tissues. So, literally, a different protein is encoded. So, even at that level, the level of the central dogma, it doesn't mean the central dogma is wrong, but it does mean that it needs to be modified and, made more sophisticated. Um, but I suggest in the book that the way I think about the genome now is that, and I should say the genome rather than genes, because there's not even a clear distinction anymore between which bits of our DNA are can be considered genes and
Yeah.
which bits are just sort of evolutionary junk, of which there absolutely is going to be some, and which bits are something in between. And actually, there's quite a lot that's something in between. I think that the modern view of the genome makes much more fuzzy the distinction between what is functional and what isn't. I kind of think of the genome now as encoding kind of adventitious matter, molecules that might have a function, that don't have a well-defined function, but that might be useful to the cell. Tiny little proteins that are almost too small to be proteins, for example, tiny little stretches of RNA that don't encode proteins but do things themselves, but do them, in combination. And we'll talk about this more. So there's all this stuff that's coming out of the genome. And so I think the best way to think about it is it's a resource. It's a source of resources, of molecular resources that cells can use. So, some of those have well-defined functions. There are some enzymes that are well-defined in terms of their structure and their function. They have to be like this to do their job. The cells absolutely need them to do their job. So, that's quite clear. But there's a whole bunch of other stuff that is much, fuzzier, that we can't even speak about it having a single function. It seems to crop up in all sorts of places, or these molecules might sort of act collectively. So that's the way I think to think about it. It's a source of molecular resources for the cell.
Yeah. It's well defined, but I think it's sort of apparent how language just fails at biology. Does like it's so hard to encapsulate biology in language, because biology is this fuzzy, chaotic, and oftentimes random system of continuums and spectrums, and language is all about labels and discrete chunks, and this is this and this is that, and it just feels such a poor medium of describing biology. Like, we almost need to add another dimension to it. It needs like a 3D animation or a world or virtual reality or something to really get at the essence. Because just any linear sentence you use to describe biological processes, you can always point holes in and say, well, actually, here's an exception here, and it doesn't really work like that in this case. And just, you can't really get that fuzziness. And it's why I appreciate how well a job you do at it in writing, because it's just so difficult. I mean, have you found that you thought that just the sentences or just the language just doesn't really seem to get at it sometimes?
Yeah, absolutely that. And again, that's why I wanted the book to start a discussion, or, maybe it's already happening, to contribute to the discussion of what narratives we need to describe biology now. If we recognize, as I think we should, that the old sort of computational linear narrative is no longer the right one, how else do we talk about this? Because we absolutely need narratives to
Yes.
understand biology, or to get some foothold on it. None of them is going to be perfect, but we need them to do some kind of job. So yeah, it's certainly true that language is a problem, but I think what has tended to happen in the past is that, for that reason, and also because this is sort of a way that our minds work, we have fallen back on metaphors and analogies from our technologies. So,
Yes.
and this is something that's always happened for the body as a whole. I mean, this goes right back to the sort of mechanistic philosophy of Descartes, and in the 17th century, that he thought of the body as a machine, and we have continually thought about it as a machine. But the nature of that machine has changed, whether it's sort of hydraulic, or whether it's computational now, or whether it's mechanical. We talk about it in machine terms. And the one thing I really wanted to stress in the book is that isn't going to do the job in biology. There are occasions, there are some proteins, for example, that really do look like machines, and we can use machine-like terms to understand them. There are proteins that look like sort of rotary motors, and we can think about them in terms of torque and,
Yes.
those sorts of concepts. And that's fine. Most proteins aren't like that. We talk about them as molecular machines, but actually they don't behave anything like our machines do. They're much too fluid and stochastic and all the rest of it. But the body as a whole is nothing like a machine. We don't, machines tend to be made from parts that have particular functions, and they're there to do a job. And again, that's not in general the case in biology. It's sometimes the case, but some of the most important bits of us at the molecular scale and upwards aren't bits that we can say, oh, this protein does this. It's this sort of protein. We find them popping up in several or maybe many different contexts. So, what they do depends on their context. And that isn't a principle that we generally find in machines. So whatever language we use, I think we need to drag it away from this idea that, we talk about it in terms of mechanism and machine.
Yeah. Yeah, I think the animations that we do have as well definitely paint the proteins in that machinery-like way. Like you see the flagella of the bacterium, which it literally looks like it was constructed in an engineer's lab and just like plugged onto a bacterium, which just is this big motor. And like the ATPase, which is like cranking different enzymes together. And they really do look machine-like in the way that we've animated it together, because again, it's just like extremely hard to do it any other way. I guess it's hard, you can't encapsulate that fuzziness and randomness in, and the dynesin, the kinase, when they're dragging molecules down actin filaments, and like all these animations that we have are just so machine-like because it's just so hard to do it any other way.
Yeah, it is. And I understand that. And, a lot of those animations are ones of molecules of proteins that are more machine-like. So
Yeah.
these motor proteins, they are a little bit, we call them motor proteins. There's a metaphor, and it's not an unreasonable one.
Yeah.
But, many proteins aren't working that way at all. And those animations, I totally understand why you have to simplify them, why you have to do something like this. But it does make me smile when you see proteins kind of zooming in like little,
Homing beacons,
hovercraft
right? Yeah.
or drones or something, guided to their target,
Yes.
as though there's some force that's sort of pulling them there. And that is not, how the molecular world works. Of course, it's totally diffusional. And not only that, you see them as though this is happening in some big open space where things can fly around. Whereas, we now have good snapshots of what the cells really look like, and it's chaos in that.
Yes.
They're absolutely jammed with molecules sort of, bumping into one another. So you couldn't possibly do an animation that would make sense of a picture like that. So, I understand that, but it does give us the wrong impression of not only how these things work, but what is the nature of the kind of process, we're talking about, which cannot be machine-like in a place that is so stochastic, so noisy as living
Yes.
things are.
Yeah. Are genes selfish?
I think that, there's a section in the book that critiques the notion of a selfish gene, but I wanted to come at it from the point of view of thinking, well, look, this has been a useful way for some biologists to think about what genes do. So let's not throw the baby out with the bathwater. And the way I kind of think about it now is calling genes selfish is not saying anything about the nature of genes themselves, nothing whatsoever. Obviously it's metaphorical, but even as a metaphor, it doesn't work for what genes actually do day to day in our bodies. It's a metaphor for describing the model that many evolutionary population geneticists use to think about genes. So that's really what it is. It's not it's not a metaphor about genes themselves or about reality. It's a metaphor to capture that particular model, which is a model that can be useful if we're wanting to understand how different variants of genes spread through a population. And we can do that without any reference to the things that are actually alive, living organisms. In fact, when we do that in those models, the organisms become a puzzle. They become what Richard Dawkins called, there's a paradox of the organism in that model, because you start to think, well, why are organisms needed at all? And the best you can do, and this is what Richard has famously talked about, is to say, well, then the organism is just a vehicle, just a passive vehicle for the genes, and it's the genes that are calling all the shots.
Yes.
In that model, that is how you think about it. If you then make the mistake of thinking you are talking about actual living things, about how biology actually works, at the molecular scale or at the organismal scale, then you've lost sight of what you're actually talking about. You've lost sight of the fact that you're talking about the model. So if genes are selfish at all, let's confine that to that particular arena of evolutionary population genetics. There is no meaningful way in which the way genes function in living systems can be considered selfish. There are actually bits of our genome that I think you could reasonably describe as selfish. These are things like transposons, these elements that are just exist to kind of make copies of themselves within the genome. And if they were allowed to do that, if they were given free rein to do that, they're often sort of bits of viruses that have made their way into our genome. And if they were allowed to do that unchecked, they genuinely would be selfish in the sense, because this is really what selfishness is about. It's not just about looking out for yourself. It's looking out for yourself at the expense of everyone else.
Yeah.
And if they were allowed to do that unchecked, the genome would kind of fall apart pretty quickly. It would just stop working. And that is precisely why we have very sophisticated molecular sort of immune systems within the cell nucleus to keep those transposons under control. So the notion of there being selfish bits of DNA is a perfectly good one. And in fact, it was one that predates the Richard Dawkins selfish gene. That's another of the worries I have about it, because there was a perfectly good, sensible way to talk about selfish DNA that became muddied once you said, well, actually all genes are like this.
Yeah.
We just need to be very clear what we're talking about when we talk about things like selfish genes.
For sure.
The Giant Shoulder mission is to explore radical ideas in biology, neuroscience, and consciousness, and elevate those stories to the highest possible level while keeping them accessible to everyone. If this interests you and you want to support independent science, then please consider subscribing to the Clips channel. Check out our 26 Neuroscience book. You can download it for free